Okay, this video is to discuss the somewhat confusing histology of the retina. So what you should have already done is gone through the videos talking about the eye, gone through the video of the eye dissection and seen all the different pieces. Now what we're going to do is we're going to focus just on this one layer right here, the retina, and go up microscopically to look at the retina in a histology type fashion. And when we look at the retina, there are a lot of different layers to capture. So a lot of different layers, and actually 3 sets of cells, so 3 layers of cells that we're going to be looking at. So we'll be doing the histological slide of the retina and capturing all of the important cells and structures that you're going to find here. So the first thing that we're going to do is we're going to draw the bottom layer, the base layer. I'm going to do this in, let's go ahead and do red here. So this bottom layer is going to be the choroid coat. So this is basically where all of the blood vessels, the vasculature that supports the retina is. So this is where all the oxygen and CO2 exchange happens. This is basically going more superficial or outside of the inside part of the eyeball. So very far away from the vitreous humors and the other stuff that's going on in the middle of the eyeball. So we've got our choroid coat, and then right on top of the choroid coat is a single thin layer in black here, and that's going to be our pigmented layer. Right, so this part out here is the sclera and superficial eyeball, whereas this part over here would be the deep eyeball, or the vitreous humors area. So our first layer here is the choroid coat. So from the outside of the eyeball, the sclera towards the middle, we get the choroid coat. And this black layer right here is going to be the pigmented layer. Then the next thing that we run into are the rods and cones. So the rods and cones we can graphically depict here. Don't need to do a ton of them. There's some rods. Why don't we do some cone action going on? Something like that. The next layer that we come to is the layer of rods and cones. Now these rods and cones are only part of a cell, so they're actually connected to another portion of the cell. They're a piece of the cell that sort of projects out, and so they're going to have nuclei right here. We'll do green for the cone nuclei. So we are basically going to be having a layer. I don't think I can copy and paste those. Anyway, we're going to be having a layer of cones and rods, and then a layer of nuclei of the cones and rods. So the nuclei layer of cones and rods. Now these cones and rods are what actually pick up the light and transform that light into electrical signals. There's going to be another set of cells that we'll do in blue here that we'll do in blue that have nuclei here, and these are called bipolar cells. So these are neurons. They have an extension that's going to interact with the rod cells here. Remember, bipolar neurons have 2 projections. One are the dendrites that interact with the cell. The other side is the axons that'll send that information away. So we've got our bipolar layers here. And then the cells, there's also cells in here called horizontal cells, but they're almost impossible to differentiate from the bipolar nuclei, so don't worry about being able to find them on a histology slide. So our bipolar nuclei are then going to be interacting with the last of our cells, which can be found here. And these are ganglion cells. So a ganglion nuclei layer. These ganglion nuclei will be interacting with the bipolars, and they're going to send out very long axons that will eventually go into that optic disc and go backwards as cranial nerve number 2, the optic nerve. So all of these axons will eventually go out and mix together and create the optic nerve, which will go back into the brain and connect to the occipital lobe, giving us vision, basically. So this is the ganglion cell layer. Now, what makes this histology really weird to think about is where light is coming from. So light needs to hit the rods and the cones in order to be transformed or transferred into electrical signals that can then be passed to the ganglion cells and taken out through the nerve. Now, you would think light would come this direction and hit the rods and cones, but in fact light is coming through the pupil, through the lens, through the vitreous humors, this way. So this is the pathway of light. And in fact, light would have to go through our ganglion cell layer. It'd have to go through our bipolar nuclei cell layer, even go through the nuclei of our rods and cones before it finally hits this layer right here, before it finally hits the rod and cone layer. So how does it do that? How does light go through all of these cells and not get distorted? Well, we have to remember when we're looking histologically, the cells have been prepared. They've been stained with various kinds of chemicals that make them look pink or purple or red or something like that. In your body, naturally, many of these cells are just clear, transparent. So light would be able to travel through all of them and finally hit the rod and cone layer here. So a picture something like this, including the pathway of light here, I would like. Oops, that's ugly. There we go. And just to see what that looks like histologically. So this would be our drawing. What would it look like histologically? If we go back to here and we see the eye, this layer down here would be the choroid coat layer. So our red layer right there. So here's the choroid coat with big blood vessels bringing oxygen and taking away CO2. This black line right here, that would be our black line right there, the pigmented layer. This area right here would be the projections of our rod and cone cells, so the actual rods and the actual cones that light will hit and will be transformed into electrical signals. Now that's just part of the cell. The actual rods and cones are connected to the nuclei of all of those individual cells, which can be found in this layer right here. So this layer right here, this is our rod and cone layer right there. This group of nuclei right here are these nuclei right here of the rods and cones. And then our rods and cones are collecting the light, and that information needs to be sent on to the ganglion cells, but there is a connecting cell in between that, and that's all of these many bipolar nuclei here. So the bipolar nuclei have two extensions, one that go down and connect to these guys, and the same cell would have an extension that comes up and connects to our ganglion cells up here. So this layer of nuclei is the layer of our bipolar nuclei. And then this is where the axon part of the bipolars would be until they connect up to ganglion cells up here. So that would be this layer right here, our ganglion nuclei layer. The ganglion nuclei would then send their long axons this way towards the optic disc somewhere over here, where it then would continue as the optic nerve and go to the occipital lobe to be processed. So in this picture, light would actually be coming from here, go through this layer, through this layer, through this layer, hit this layer, get turned into electrical signals that would be passed from this cell to this cell to this cell, then out the axons into the brain. So draw a picture something like this, including those layers. So what I have just written out here, please include on your drawing exactly where those are. And yeah, have fun with it. All right, see ya.